So our third speaker, our third young scientist speaker is Augustina Fernandez Casafuz from University of Buenos Aires, Argentina. And she's gonna talk about transport and mechanical properties of mitochondria. So Augustina, are you ready?
Yes, sorry. I can't start my video.
Yeah,
right now.
yeah, I think Cynthia will open your video and then you can share the screen.
Yes. Here, now, is my video started? Sorry. I can't see me.
Yes, your video is fine. You can just put the presentation.
Sorry.
Yeah, it's in the presentation mode.
Yes. Okay, perfect. So thank you very much to all of you who are still here and to the organizers for the opportunity of telling all of you about my work. My name is Agustina and I'm doing my PhD at the University of Buenos Aires in Argentina. Sorry, I'd like to, that's it. Sorry, I have this Zoom bar. Okay. So I will tell you about some of the work I'm doing in my PhD related to mitochondria. There. So as a brief introduction, thankfully Alicia has already talked about mitochondria. What I would like to remind you all is that mitochondria are dynamic organelles. They have many functions in cells, very important ones, like for example, energy production, ATP production. And what I'm most interested about is that because of this, their cellular localization is very important. No? They need to be precisely located in cells in order to have a correct functioning. And also, they are very dynamic organelles. This Alicia talked about this a lot. So we have mitochondria that can have many shapes and sizes, different ones. And so in order to achieve this, this correct localization, the cells have this active transport system, which consists of molecular motors carrying different cargoes. This can be mitochondria or other organelles along filaments of the cytoskeleton. These proteins, these molecular motors, are some specific proteins that can attach both to the cargo and they bind to the, for example, the microtubule. And through the hydrolysis of ATP, they are walking like this with these little feet, and they carry cargoes. In particular, we are interested in transport along microtubules only. So in general, we use a drug called latronculin, which depolymerizes all of the actin filaments. And so what we are seeing here is microtubule transport, which is mediated only by two of the motor families, namely kinesin and dynein. Kinesins usually carry cargoes to the cell periphery, and dyneins do the opposite. They carry cargoes towards the cell center. And so even though here it is illustrated very simplistically, and you can have a lot of motors working at the same time on the same cargo. And so it is very difficult to know whether if it moves this way or the other way. And so this is what we are trying to understand, this bidirectional transport. In this way, some previous work made by Cecilia De Rossi, she was studying mitochondria in Xenopus levis melanophore cells in vivo. There she analyzed these confocal microscopy images where you can see mitochondria in red and microtubules in green. And what she saw is that even though mitochondria have a lot of different shapes, most of them prefer to be in a rod-like shape to be transported, and also that because of this elongated shape, they can also retract and extend during transport. No? They change their length during transport, they can do that. And in this line is where I started my work. I will tell you now about a few results of a model we proposed and was published last year. So what we do is we have these experimental images where we use a single particle tracking routine. We take each mitochondrion individually and we follow its center of mass to recover its trajectory in time. Now we can see here that sometimes they move in one direction and then they go back because of these two kinds of motors that carry them along the microtubules. But also we can calculate their length during the transport, and we see that this length fluctuates, just as Ceci proposed when she saw that. And what we found is that the mitochondria that are retracting and extending, they do that in 20 to 30% of their initial length. But what's more interesting is that we saw there is a correlation between the length, the speed of the mitochondrion, and whether if it extends or retracts, or there are some that don't change appreciably their length. And so we see that mitochondria that are extending, they have a lower mean speed. And so this is what we are trying to understand. There are a lot of questions that arises from this, like why does these organelles behave like this? Why are they extended? Which are the forces involved in this deformation? If they are elongated, why do they retract or extend? And so in order to do this, we proposed a mechanical model. In literature, there are a lot of models, of course, of intracellular transport, but most of them usually work with very round or rigid or punctual organelles, so they can't really explain. We cannot really use them to explain what happens to mitochondria. So we did what any physicist would do. We proposed that the cargon is now a harmonic oscillator, a damped harmonic oscillator. So now instead of having one round cargo, we have these two nodes coupled, and so we gain this flexibility. Now the cargo can elongate and can shorten. But also what we gain is that these two nodes are like natural anchoring points for the motors. So the motors that I told you, they are bound to mitochondria and they carry them along the microtubule. Here, they can only bind to one of the two nodes, and so the forces they exert are local. This is a very simplistic model, but it can be easily extended to other systems, and so we are very happy because we can explain a lot of things with this very simplistic model. So what we do is we solve two coupled Langevin equations for the cargo, for the mitochondria, and then we also have an independent motor dynamics. As I told you, you can have a lot of motors working at the same time on the cargo, but each of them has an independent dynamic. At first, motors can diffuse all along the cargo when they are unbound from the microtubule, but at some point with probability PA, they can bind the cargo to the microtubule in an elastic way. But as I told you, they can do so only on the two nodes, on the front or on the back. And once they are bound, they can take a step in their direction of motion with probability PS. And doing so, they exert a force on this node particularly. And then, well, they can take a lot of steps, they can take only one, and they also can detach from the microtubule with a probability PD. Both of these probabilities depend on the force, the opposing force the motor is acting. So if they are acting against a large force, they will probably detach more easily, or they will stay still and not walk a lot. And so this is a cycle that repeats. And in these simulations, what we can recover is the cargo speed and the length variation, which are the same observables we have from the experiments. But also here we have the motor distribution, because we know exactly where each of the motors are at each simulation step. And so with this, we analyze two distinct scenarios for the motors. One in which we consider the cargo being carried by one team of all identical motors. We can vary the number of motors in the team, but they are all the same. They all go to the same direction. And then one, when we have a high competitivity, we have both of the motors, both of the polarity motors acting at the same time. And so we have what we call a tug-of-war scenario. In the first case, when we have an identical motors team, what we can see here is the trajectory of the cargo being carried by in the light blue, by a 2-motor team, and in darker blue, by a 10-motor team. And of course, as expected, we can see here that the speed is greater as the number of motors grows, and also the processivity. But what is more interesting is that when we look at the deformation, this is the change in length of the mitochondria of the cargo relative to its natural length, the one of the spring, we see that we have periods of stretching and retractions, but most of the time this cargo prefers to be in a constricted state. It is negative, and so we wonder, why does this happen? It is here that we take advantage of the simulations and we analyze how do the motors distribute. What are the motors doing that the cargo tends to be in this shortened state? And so here we can see a probability density estimate of the position of the motors along the cargo. And we can see here in purple, this curve here in purple shows the distribution for the motors when they are inactive. This means that they cannot mount to the microtubule, they are only diffusing all along, and so we have this homogeneous density. But when we have motors that can bind to the microtubule, remember they can only bind to the nodes that are on the front and on the back of the cargo. We have these peaks. They tend to locate on the peaks of the nodes, and we see that as the number of motors increases in the team, they tend to locate mostly on the back part. This is a really asymmetrical distribution, and so what we have is something like this. When we have, for example, 2 motors, they are either at the front or the back, and it's the same, they have the same probability. But when we have, for example, 3, 5, or 10 kinesins, we see that they locate at the back of the organelle. Thus, they are pushing the organelle, having the rear edge larger speed, and so it tends to contract like this, like we saw on the deformation plot. Then, if we look on the other hand, on the tug-of-war scenario where we have both of the motors, we have the kinesins going to the right and linings going to the left, what we see on the trajectory is that we have this bidirectional transport. We have that at some points it goes to one direction and then it reverses and goes in the opposite direction. But the mean speed of this is actually really, really small. Now, this depends obviously on if it is a very highly competitive team, 10 versus 10, rather than, for example, 10 versus 5, and then you have a mean speed going to the direction of the more numerous team. But as we look to the deformation, we see here that even though you have the periods of stretching and retracting, it prefers to be in an elongated state, the cargo. So now you have that the cargo behaves in a very different way. Now it's extended more likely. And again, as we go to the motor distribution, for example, now if we see this from the team of kinesins perspective, for example, now in blue we have, well, they are only the kinesins, they are walking, they have no force opposing them, and so they locate on the back. But as we add more members to the opposite team, now they are competing with the other team. They have like this opposing force, and so they shift their location. They change from the back to the front of the cargo. We have this transition from pushing motors to pulling motors. And in doing so, the same obviously happens with dynamics. They are pulling, but from what is their end now, because they're moving to the other side. And so we have that the cargo tends to extend. And with this, what I would like for you to take home is that these simulations allow us to explore different scenarios for the intracellular transport, and thus we gain a deeper understanding of the mechanisms underlying the intracellular transport. Now, in this particular case, for example, we can see that we can infer that mitochondria that extend and have a lower mean speed are probably in a very highly competitive scenario with both of the motors acting at the same time. No, so you have these extended organelles that in average don't move, whereas when you have these mitochondria that have a large speed and they are retracting, you can think that they're probably being carried by one team, or in a less competitive team scenario. Okay, so that will be all. I would like to thank you all again, especially here Luciana and Ceci. Without them, this work wouldn't happen. And of course, if you have any doubts or any questions, you can ask them here, or here is my email address for any contact you would like to say. Thank you.
Thank you, Augustina. So this moving mitochondria is also new for me. But I think people here will have specific questions for you. So the paper is open for questions.
Yes.
May I?
Yeah, please, Gonzalo.
Sí.
Yes. Hello.
Many thanks, Agustina. Very nice talk. I really enjoyed it. I have some questions related to, for instance, if you know how many sites you have for kinesin and dynein per mitochondria, and if they are all in a particular place or another, and how many microtubules are surrounding one particular mitochondria. That will be just...
Do you mean in the experimental setup, I mean in vivo in the cells, or
vivo,
in investigation?
Yeah. At least in the Xenopus levis that you've been studying.
No, as I said, well, I didn't say it, but you have something like this. So this is the movie. What we are doing is we are taking this in the far end of these cells. So we know it's a 2D motion because they are very thin cells. But obviously we are seeing things like this, like in one microtubule at a time. Sometimes there are more than one microtubule. For us, it's not important, at least for now, on which microtubule they are moving. But regarding the number of motors, that is something we cannot access so easily experimentally. There is one work that I didn't mention because of lack of time that Ceci did, but it requires a fluorescence spectroscopy correlation. And right. Here what they saw, it is in green, is the kinesins. And so this is like the most you can access the motor dynamics in vivo. This is a very complicated experiment, and the results are also very, very complicated to tell. I didn't do anything of this, so I can't really tell you, but we don't really know how many motors are acting at the same time, but we do know that they are, for I think in literature there are a lot of, I don't remember the name, but if you take and you see like in SEM or in these electron microscopy movies, you see that they are stuck to the mitochondria. There are a lot of them, maybe. You don't know if they are acting or not, so it is very complicated.
Mm-hmm. Can I continue? Because Mauricio, and I don't know if there's anyone else.
Yeah, there is other people, but you can ask one more question, then I...
Okay, so...
We have time, I think. Yeah.
Okay, thank you, Mauricio. I didn't want to spoil the party.
No, I think there'll be plenty of time for the party.
Okay, so the issue is, Agustina, that I was thinking that maybe when you have most dynein or more kinesin, you are prone to go more to the periphery or to the middle of the cell.
Yes.
So am I correct? And then, for instance, that could have some relevance during cell division, especially when you have oocytes.
Yes.
Right? And you are going to have the divisions in during the zygote and those stages. So this could be particularly relevant in those stages. So what's your idea about it?
Yes, I think you're right. We haven't studied this in any particular cellular state. I think maybe in the future it would be very interesting to see what happens during cell division, because of what you're saying. It is very important to really localize the mitochondria. And there are some works that that deal with this, and at some points how this is really necessary to really mix up in a sort of way all the mitochondria in order for the daughter cells to have the same, and if you have some defectuous, or I don't know how to say it, sorry, mitochondria, that they are end up in the same cell, and those sorts of things, it is very important, the transport there. Yes.
Mm-hmm. But the kinesin is going to more towards the center or the periphery in general? The orientation you have here.
Yes, in general, they go to the plus end of the microtubule, which is usually the periphery. And
the periphery, right? Yeah. Yeah. Okay. Thank you. Thanks a lot, Mauricio.
Thank you.
You're welcome, Gonzalo. Thank you. Now, please, Alicia, make your question.
So first, thank you for that talk. That was fascinating. I thought it was amazing. And looking at this from a very different perspective of most groups, I think you can do these simulations with many different cell types and different kinds of characteristics of dynamics. That's going to be really interesting. Yeah. I was wondering, and this is kind of crazy, as if you hadn't done enough work already, but can you simulate what the metabolic cost of all this movement is? In other words, we know the structures and the function of these molecular motors, and we know how much these mitochondria are moving. Can we do simulations that estimate how much ATP this costs a cell? Because this is something I think of a lot. The cells do this, it must be worth it. But it looks like there's a lot of energy involved. And I'm just wondering if you can think up that kind of level of simulation on top of what you already have.
Yes, I get where you're going. I in particular, I don't do those kind of simulations, but I think there are many models that, well, they are not about mitochondria. They're what I said about more rounded cargos, but they do these calculations on ATP concentration and how all of these parameters change and how it affects the transport. I think there are many works that to deal with that. In particular, we use more of a stochastic approach, where we have these probabilities that they are drawn from the literature, but they are not. I don't calculate them in terms of ATP or anything. But yes,
cool. That's really nice data. Thank you for that.
No, thank you.
Thank you, Alicia. Thank you, Augustina. Antonio has a question as well.
Oh, yeah. Thank you, Agustin, for such a nice talk. So, very interesting model. So, first, have you tried to correlate or to interpret? Well, you simulate, you have the position a long time. So that's, of course, related to the velocity. So is there any pattern in the velocity of yours? Can you correlate the velocity of your simulation with the pattern of going backward or forward in the mitochondria? And second question is, how general? To me, it seems that your simulations are quite general, right? To me, it doesn't really matter whether it is a mitochondria or any other sort of vesicle or nanoparticle or whatever. So do you think you could correlate the variations in length to the stiffness of whatever is being transported? Like if you change the conditions of the sample in the cell, is for example, stress scenario, and then you get, a stiffer, vesicle somehow, and could you just like relate stiffness with the pattern of variation in length?
Yes, I will answer first the second question. Yes, of course, this is a very general simulation. In the simulation, it doesn't matter if it is a mitochondrion or another type of organelle. But for us, this is more of a motivation because we have these experimental results on mitochondria. And so we know that these are very dynamic organelles and they change sizes. And so we apply this model to them, but it can easily be extended to other type of organelles, like you say, by changing the stiffness of the spring. So we can have peroxisons or anything we would like. We can make it more round or more elongated, and so we can do this.
Can I just add? Could you do the opposite? I mean, you measure either the variations in length or the velocity, and then you simulate the data, the experimental data, and then you determine the distribution of the motors, whether it's like one motor, two types of motor, and whether this will go backward or forward. I mean, can you do the opposite? No, you have the experimental data and you simulate it. And then from that simulation, you get a parameter, to say, oh, this mitochondria or whatever is now, connected to the microtubule through two types or those different types of motors. And there are more motors in this part of the vesicle or in that part of the membrane bound organelle. I'm trying to correlate your model that you showed you worked with the data you have to get some connections with the experiments and see if we can get some information about the distribution of the motors and the structure, the rheological properties of the vesicle.
Yes, it's kind of a far-reach thing to do that. We would like to do that. I don't know if I have here because I talked about these results that Ceci had previous to our model, when I said that the mitochondria retracted and extended, and she actually found a correlation between the direction of motion of mitochondria. I don't know if I put it here. No, I didn't. So in the previous work, she found that mitochondria that retracted were mostly being carried to the cell center by dyneins, because of the direction, and mitochondria that she found very few ones that extended, and the other ones, the ones that were going to the periphery, they kind of didn't change their size. And so, but this is again like far, I don't know how to say it, like a wild guess. Now, what we saw here, well, in these results, we are not comparing the direction of motion in these experimental results, but those of her, yes. But what we can do in light of those previous results is that we can say, of course, the ones that are extending, and so they are probably not moving at all because they are going back and forward all the time. They are mostly still. They are most likely being carried by both types of motors are acting at the same time. But then you see here that we have the ones that don't change and the ones that retract, and they are both have a larger speed. And considering what she saw, the ones that retracted are probably being carried by a numerous team of dyneins. And the ones that don't change their length, what we can assume is that they are being carried by a team of kinesins because of the direction of motion she established. And because what we saw here, that I didn't spend too much time, is that when we have two kinesins, they can be either at both of the sides. So if we have a few motors, it's more probably that they are going anywhere in the mitochondria. And here we can see in the light blue in the deformation that it doesn't really change its size. It has sometimes when it goes, when it retracts and then it extends, but in average, the length doesn't vary enough. No, I don't know if that answers a bit your question, but I think that is as far as we can go for these types of simulations, because it is experimentally it's very difficult to know which of the motors are acting because of what I said earlier about the technical difficulties. The motors are very, very small. You can see them through this fluorescence microscopy, spectroscopy, but it is really difficult. Well, what she also saw here is that there is, I'm sorry that they are in Spanish, but I'm more interested in the picture. What she saw in this kind of experiment with the fluorescence spectroscopy correlation is that motors spend different time being when they are located in the back. These are all kinesins when they are in the back than when they are in the front. So I think that's, we don't know how many are acting at the same time, but they are spending different amounts of time. The ones in the back have a faster exchange than those on the leading edge. Well, if you are interested,
I can give you a suggestion on where to apply your models if you are interested in that. So this is very similar to what we find when we are looking at enzymes that degrade the cellulose. You know, so you have this bagasse from cellulose, so you have these long polymers, and you have this, you know, complex of enzymes that degrade the cellulose and break them down in small pieces. So they kind of do the same sort of movement. So it might be a good, you know, model system for you to get experimental data on. And people will be interested in, you know, that's the way they do. They use FCS to produce data on the dynamics of this, how these enzymes, you know, these catalytic enzymes, how they move on the fiber to degrade it. So it's pretty much the same situation there. So, well, it's not in the cell, but it could be an application of the model you have there. And it will have some, you know, let's say, economical impact in principle.
Okay, thank you very much.
Yeah, thank you.
The senior scientist became very interested in your work. Congratulations, Augustina.
Thank you. Thank you.
Before I end, I would like to share the screen of our next webinar. So I also would like to announce our next webinar, which will be in August 18th. And we'll have the presence of the Washu, which some of us already have attended his speech. It's wonderful. He's going to talk about electrocryomicroscopy. And we'll also have 4 young scientists, Priscilla from South Africa, Rito Brita from India, Kamalendu from India as well, and Raissa from São Paulo. So I hope you can join us again in 15 days. And I wish you a wonderful afternoon. So with that, I thank you for participating, and bye-bye.
Bye, thanks.
Bye,
Thanks
bye.
for organizing
Hey,
all this. Bye.
Obrigado, Alicia, por ter estado com a gente, viu? Muito obrigado.
Hello,
Obrigado. Thank you, Augustine, and the others, Caroline and Eleonora. It was great.
Bye.
Bye-bye.